Hello, good evening to everyone. I welcome you all to Unacademy on the CSIR NET channel of Life Sciences. Myself Himanshu and I welcome you all. And today we will be studying structural organization and function of intercellular organisms. We will be taking the case of chloroplast. So I welcome you all in today's lecture. All those who are present, they can type hello so that I can understand that आप लोग सभी लोग online आ चुके हैं. तो बिफोर वी स्टार्ट हम लोग शुरू करने से पहले मैं आपको ये बताना चाहूँगा कि आप गूगल प्ले स्टोर पे जाएं और वहाँ पर अनअकैडमी का लर्निंग ऐप आपको मिलेगा, उसी को आप इंस्टॉल करें एंड देन यू नीड टू सेलेक्ट योर गोल. इन द सेलेक्ट योर गोल यू नीड टू सेलेक्ट CSIR UGC नेट कैटेगरी. तो जैसे ही आप CSIR UGC नेट कैटेगरी को चूज़ करेंगे तो आपको वेरियस एजुकेटर्स के टॉप फ्री कोर्सेज आपको एक्सेस के लिए मिल जाएंगे. एंड इफ़ यू वांट द प्लस एक्सपीरियंस, if you want to go for the plus if you want to go for the exclusive guidance and exclusive coverage, you need to subscribe for to the plus course. For that you will be needing CSIR UGC net subscription, and this subscription is for 1 month, 3 months, 6 months, 12 months and 24 months. I would suggest you to get a subscription for 12 months and 24 months, and you can use my code HY10. You will be getting 10% off. So the entire course can be bought for 10,395 instead of 11,550. You can do the payment either by credit card or debit card or by EMI, and these EMI are no cost EMI. You can also pay through the net banking. तो आज का सेशन होगा क्लोरोप्लास्ट के बारे में, और मैं आप सभी लोगों का स्वागत करता हूँ अनअकैडमी के CSIR नेट प्लेटफॉर्म पर. So let's begin today's class. So autotrophs, they sustain themselves without eating anything derived from other organisms. So autotrophs, they sustain themselves without eating anything derived from other organisms. Autotrophs are the producers of the biosphere, producing organic molecules from CO2 and other inorganic molecules. Almost all plants are photoautotrophs, using the energy of the sunlight to make organic molecules from H2O and CO2. तो इसमें बताया गया कि ऑटोट्रॉफ्स क्या होते हैं. So there is a basic differentiation between autotrophs, heterotrophs, or the primary producer and the primary consumers. So over here we are describing the autotrophs. They include the plant unicellular protist, cyanobacteria, purple sulfur bacteria, multicellular algae. And heterotrophs, they obtain their organic material from other organisms. They are the consumers of the biosphere. Among all heterotrophs, including humans, depend on the photoautotrophs for food and oxygen. फोटोसिंथेसिस वो प्रोसेस होता है जो कि कन्वर्ट करता है लाइट एनर्जी को केमिकल एनर्जी में, केमिकल एनर्जी ऑफ द फूड में. एंड क्लोरोप्लास्ट आर स्ट्रक्चरली सिमिलर टू एंड लाइकली इवॉल्व फ्रॉम फोटोसिंथेसिस bacteria. The structural organization of these cells allows for chemical reactions of photosynthesis. तो क्लोरोप्लास्ट का जो एवोल्यूशन हुआ है वो एक फोटोसिंथेटिक बैक्टीरिया से हुआ है, क्योंकि वो स्ट्रक्चरली सिमिलर है, ऐसा कहा जाता है. और जो स्ट्रक्चरल ऑर्गेनाइजेशन हुआ है दीज सेल्स का, दे अलाउ फॉर केमिकल रिएक्शंस ऑफ फोटोसिंथेसिस. JB Yadav, hello. I welcome you all to this platform. Chloroplasts are the sites of photosynthesis in plants. Leaves are the major locations of photosynthesis. जैसा कि हम लोग को पता है कि green color is from the chlorophyll and the green pigment is within the chloroplast. So light energy absorbed by the chlorophyll derives the synthesis of organic molecules in the chloroplast. CO2 enters and O2 exits the leaf through microscopic pores called stomata. So CO2 enters and O2 exits the leaf through microscopic pores called stomata. So chloroplasts are found mainly in cells of mesophyll, the interior tissue of the leaf. A typical mesophyll cell has 30 to 40 chloroplasts. The chlorophyll is in the membrane of thylakoid that are connected in sacs in the chloroplast, and the thylakoids may be stacked in columns called as grana. Chloroplast also contains stroma, which is a dense fluid. तो ये स्ट्रक्चर है क्लोरोप्लास्ट का. This one is the structure of the chloroplast. This is the stroma. This is the grana or the granum. This is the thylakoid, and this is the thylakoid space. This is the inner membrane. This one is the outer membrane, and this one is the intermembrane space. ये लीफ का मेसोफिल का क्रॉस सेक्शन है. तो लीफ के मेसोफिल में क्लोरोप्लास्ट होता है. This is the structure of chloroplast. This is the stroma. This one is the granum. This one is the thylakoid. This one is the thylakoid space. This one is the inner membrane. This one is the outer membrane. This one is the intermembrane space. This one is the chloroplast. And this one is the stroma. So photosynthesis reaction, it can be summarized as 6 CO2 plus 12 H2O plus light energy gives C6H12O6 plus 6 O2 plus 6 H2O. So this is the general reaction of photosynthesis. So chloroplasts, they split hydrogen into hydrogen and oxygen, incorporating the electrons of the hydrogen into the sugar molecules. So chloroplasts, they undergo the splitting of water into hydrogen and oxygen, and they incorporate the electrons of the hydrogen into the sugar molecules. तो फोटोसिंथेसिस एक रिडॉक्स प्रोसेस है जिसमें कि वाटर ऑक्सीडाइज होता है और CO2 रिड्यूस होती है. वाटर इज ऑक्सीडाइज एंड CO2 इज रिड्यूस्ड. So there are two stages of photosynthesis. So photosynthesis consists of light reaction or the photo part. It consists of the light reaction, and it also consists of the Calvin cycle, that is the synthesis part. So the light reaction, it occurs in the thylakoid. So in the light reaction, the water is split, it releases oxygen and it reduces NADP plus to NADPH, and it generates ATP from ADP by photophosphorylation. So photophosphorylation takes place, by which ATP is generated by ADP. So phosphorylation is an important process, and photosynthesis consists of two stages. One is the light reaction, other one is the Calvin cycle, in which the synthesis takes place. So in the Calvin cycle, they take place in the stroma. The Calvin cycle, it takes place in the stroma, and it forms the sugar from CO2 using ATP and NADPH. The Calvin cycle begins with carbon fixation, incorporating the CO2 into organic molecules. So Calvin cycle sugar molecule form करती है from CO2 using ATP and NADPH, and Calvin cycle begins with carbon fixation, incorporating CO2 into organic molecules. So this is where the light reaction takes place and splitting of water molecule takes place. Light आती है और NADP प्लस कन्वर्ट होता है NADPH में और ATP की सिंथेसिस होती है और ऑक्सीजन मॉलिक्यूल रिलीज होता है. और यही ATP और NADPH यूज़ होती है कैल्विन साइकिल में, जहाँ पे शुगर मॉलिक्यूल सिंथेसाइज किए जाते हैं बाय अपटेकिंग CO2. And giving off NADP plus and ADP, which is used in the light reaction. So both these reactions occur simultaneously. So the light reaction, they convert the solar energy to chemical energy of ATP and NADPH. Their thylakoids transform the light energy into chemical energy of ATP and NADPH, as we have seen already. So what is the nature of the sunlight? It is in the form of electromagnetic energy, also called as electromagnetic radiation. Like other electromagnetic energy, light travels in rhythmic waves. Wavelength is the distance between the crest of the waves, and wavelength determines the type of electromagnetic energy. So visible light consists of wavelength that produces colors we can see. So visible light consists of wavelength that produces colors that we can see. Light also behaves as though it consists of discrete particles called photons. So this one is the shorter wavelength, this one is the longer wavelength, this one is with the higher energy, and this one is with the lower energy. So visible light is from 380 नैनोमीटर टू 750 नैनोमीटर, और ये वाला स्पेक्ट्रम यूज़ होता है फॉर फोटोसिंथेसिस. So what are pigments? What are photosynthetic pigments? Pigments are substances that absorb visible light. Pigment are substances that absorb visible light. Different pigments absorb different wavelengths. Wavelengths that are not absorbed are reflected or transmitted. Leaves appear green because chlorophyll reflects and transmits green light. Spectrophotometer measures the pigment ability to absorb various wavelengths. The machine sends light through pigments and measures the fraction of light transmitted at each wavelength. ये एक एक्सपेरिमेंट है जिसमें ग्रीन लाइट और ब्लू लाइट का चेक किया गया था कि विच शोज मैक्सिमम अब्जॉर्प्शन. तो जब ग्रीन लाइट पास करी गई, सो हाई ट्रांसमिटेंस हुआ उसका और लो अब्जॉर्प्शन हुआ. रीडिंग इंडिकेट दैट chlorophyll absorbs very little green light, but in the case of blue light, reading indicates that chlorophyll absorb most of the blue light. तो ब्लू लाइट सबसे ज़्यादा अब्जॉर्ब होती है कंपेयर टू ग्रीन लाइट. एक अब्जॉर्प्शन स्पेक्ट्रम बनता है वो कि जो कि ग्राफ होता है दैट प्लॉट्स अ पिगमेंट्स लाइट अब्जॉर्प्शन वर्सेस वेवलेंथ. तो जो अब्जॉर्प्शन स्पेक्ट्रम होता है ऑफ क्लोरोफिल ए, it suggests that violet, blue and red light works best for photosynthesis. Action spectrum profiles the relative effectiveness of different wavelengths of radiation in driving a process. ये अब्जॉर्प्शन स्पेक्ट्रा का ये ग्राफ है जहाँ पे अब्जॉर्प्शन ऑफ लाइट क्लोरोप्लास्ट के पिगमेंट्स के द्वारा हो रही है. And this one is the wavelength of light. This one is chlorophyll A. This one is chlorophyll B, and these are the carotenoids. तो जो एक्शन स्पेक्ट्रम थी photosynthesis की, that was first demonstrated in 1883 by Theodor Engelmann. In his experiment, he exposed different segments of a filamentous alga to different wavelengths. Areas receiving wavelengths favorable to photosynthesis produce excess oxygen, and he used the growth of aerobic bacteria clustered along the alga as a measure of oxygen production. So chlorophyll A is the is the main photosynthetic pigment. So chlorophyll A is the main photosynthetic pigment. Accessory pigments such as chlorophyll B broaden the spectrum used for photosynthesis. Accessory pigments called carotenoid, they absorb excess light that would damage the chlorophyll. So when a pigment absorbs light, it goes from the ground state to an excited state, which is unstable. When excited electrons fall back to the ground state, photons are given off after a glow called fluorescence. If illuminated, an isolated solution of chlorophyll will fluoresce, giving off light and heat. तो photosystem क्या होता है? It consists of a reaction center complex surrounded by light harvesting complexes. Or light harvesting complexes, they are the funnel that the energy of photons to the reaction center. A primary electron acceptor in the reaction center accepts an excited electron from chlorophyll A, and the solar power transfer of an electron from chlorophyll A molecule to the primary electron acceptor is the first step of the light reaction. This one is a thylakoid space, and this is the reaction center complex. These two are the light harvesting complexes. This is one is the primary electron receptor. तो यहाँ पे एनर्जी का ट्रांसफ़र होता है और फिर एक स्पेशल पेयर ऑफ क्लोरोफिल ए मॉलिक्यूल्स है जिसमें ये एनर्जी ट्रांसफ़र होती है, फिर वहाँ से रिएक्शन सेंटर कॉम्प्लेक्स में ट्रांसफ़र होती है. These are the pigment molecules. So this consists of a photosystem. There are two types of photosystems in the thylakoid membrane. One is photosystem 2 that functions first and is best at absorbing a wavelength of 680 nanometer. The reaction center of chlorophyll A of PS2 is called P680. So the reaction center of chlorophyll A molecule of photosystem 2 is called P680. Photosystem 1 is best at absorbing a wavelength of 700 nanometer. So the reaction center of chlorophyll A of PS1 is called P700. So during the light reactions, there are two possible routes for electron flow. One is cyclic, another one is linear. So linear electron flow, the primary pathway involves both photosystems and produces ATP and NADPH using light energy. A photon hits a pigment and its energy is passed among the pigment molecule until it excites P680. An excited electron from P680 is transferred to the primary electron acceptor. तो ये फोटो सिस्टम 2 है, जहाँ पे प्राइमरी इलेक्ट्रॉन एक्सेप्टर जो कि मेन रिएक्शन सेंटर है उसको P680 कहते हैं. These are the pigment molecule. And P680 plus is a very strong oxidizing agent. So water is split by the enzymes and the electrons are transferred from the hydrogen atom to P680 plus, thus reducing it to P680. So oxygen is released as a byproduct of this reaction. तो जैसा कि आपने देखा यहाँ स्प्लिटिंग ऑफ वाटर मॉलिक्यूल हो गया और ऑक्सीजन यहाँ से रिलीज हो गया, +2 हाइड्रोजन आयंस रिलीज हो गए और P680 प्लस कन्वर्ट हो गया P680 में. So each electron falls down an electron transport chain from the primary electron accepted of the PS2 to PS1. So energy that is released by the fall drives the creation of proton gradient across the thylakoid membrane. So diffusion of hydrogen ions across the membrane drives ATP synthesis. तो यहाँ ये P680 का प्राइमरी इलेक्ट्रॉन एक्सेप्टर उसके बाद प्लास्टोक्विनोन में जाता है, फिर साइटोक्रोम कॉम्प्लेक्स में जाता है जो कि एक इलेक्ट्रॉन ट्रांसपोर्ट चेन का पार्ट है, और प्लास्टोसाइनिन में जाता है जहाँ से ये ATP सिंथेसिस हो जाती है. In PS1 like PS2, light energy excites P700 which loses an electron to an electron acceptor. P700 plus accepts an electron passed down from PS2 via the electron transport chain. This one is the plastoquinone, this one is the plastocyanin. This one is the photosystem 1. This one is the photosystem 2. So each electron falls down an electron transport chain from the primary electron acceptor of photosystem 1 to the protein ferredoxin. The electrons are then transferred to NADP plus and reduce it to NADPH. The electrons of NADPH are available for the reactions of the Calvin cycle. तो इस प्राइमरी इलेक्ट्रॉन एक्सेप्टर से फेरेडॉक्सिन के पास जाते हैं इलेक्ट्रॉन्स, जहाँ पे NADP प्लस रिडक्टेज एक्ट करता है और NADP प्लस का कन्वर्जन हो जाता है NADPH में. तो NADPH is formed. This all takes place in the electron. Ferredoxin is present in the electron transport chain. So cyclic electron flow uses only one photosystem and produces ATP but not NADPH. Cyclic electron flow generates surplus ATP, satisfying the higher demand in the Calvin cycle. तो ये एक साइक्लिक इलेक्ट्रॉन फ्लो है जहाँ पे केवल एटीपी का सिंथेसिस होता है, और ये प्लास्टोसाइनिन के बाद ये फोटो सिस्टम वन में जाके प्राइमरी इलेक्ट्रॉन रिसेप्टर के बाद ये फेराडॉक्सिन के बाद आके वापस साइटोक्रोम कॉम्प्लेक्स में आ जाता है, जहाँ से एटीपी की सिंथेसिस हो जाती है। इसमें केवल एटीपी की सिंथेसिस होती है और NADPH प्लस की सिंथेसिस नहीं होती है। सो दिस इज़ एन इम्पॉर्टेंट पॉइंट टू नोट इन केस ऑफ साइक्लिक फोटोफॉस्फोराइलेशन। अर्पिता तिवारी सर, आप शुरू से चैप्टर पढ़ा रहे हैं? यस, मैं जो स्टार्टिंग वाले मॉड्यूल है वहाँ से मैंने स्टार्ट किया है, और ये सीरीज़ अभी ऐसे ही चलती चली जाएगी। अभी हम लोग मॉड्यूल 1 पे हैं, मॉड्यूल 2 पे इनफैक्ट। मैंने अपना स्ट्रैटेजी वाले वीडियो में मैंने बताया था कि हाउ वी आर प्रोसीडिंग। So some organisms such as purple sulfur bacteria have photosystem 1 but not photosystem 2, and cyclic electron flow is thought to have evolved before linear electron flow. Cyclic electron flow may protect cells from light induced damage. So here is a comparison of chemoisomosis in chemiisomosis in chloroplast and mitochondria. So chloroplast and mitochondria generate ATP by chemiosmosis but uses different sources of energy. Mitochondria transfers the chemical energy from food to ATP, and chloroplast they transform the light energy into chemical energy of ATP. So spatial organization of chemoosmosis differs between the chloroplast and mitochondria but also shows similarity. In the case of mitochondria, the protons are pumped to the intermembrane space and drive the ATP synthesis as it diffuses back into the mitochondrial matrix. In chloroplast, protons are pumped into the thylakoid space and drive ATP synthesis as they diffuse back into the stroma. तो अगर आप लोग को कोई डाउट हो तो आप लोग कमेंट बॉक्स में पोस्ट कर सकते हैं। अगर आप लोग को कोई भी डाउट्स हैं तो यू कैन पोस्ट इट इन द कमेंट बॉक्स। तो ये कंपैरिजन है बिटवीन द माइटोकॉन्ड्रिया एंड द क्लोरोप्लास्ट। कैसे एटीपी सिंथेसिस होता है। So over here are the higher concentration of hydrogen ion, and over here there are lower concentration of hydrogen ion. Over here यहाँ पे lower concentration है hydrogen ions का। तो ये इनर इंटरमेम्ब्रेन स्पेस है, और ये क्लोरोप्लास्ट का स्ट्रक्चर है, जो कि जहाँ पे डिफ्यूजन से थैलाकोइड स्पेस से थैलाकोइड मेम्ब्रेन में स्ट्रोमा में जाके एटीपी सिंथेसिस हो जाती है। और इन केस ऑफ माइटोकॉन्ड्रिया ये इलेक्ट्रॉन ट्रांसपोर्ट चेन से हाइड्रोजन का ट्रांसफ़र होता है, जो कि फिर बाद में जाके डिफ्यूज़ होकर एटीपी सिंथेसिस फॉर्मेशन करता है। तो दोनों में ये डिफरेंसेस है और ये सिमिलैरिटीज़ है। So ATP and NADPH are produced on the side facing the stroma where Calvin cycle takes place. So in summary, light reactions generate the ATP and increase the potential energy of the electrons by moving them from hydrogen to NADPH. तो लाइट रिएक्शन जनरेट करते हैं एटीपी को और इंक्रीज कर देते हैं पोटेंशियल एनर्जी ऑफ इलेक्ट्रॉन्स बाय मूविंग देम फ्रॉम वाटर टू NADPH। तो ये क्लियर डायग्राम है। This one is the stroma where low concentration of hydrogen ions are there. This one is the thylakoid space where there is a high concentration of hydrogen ion, and this is the thylakoid membrane. This one again is the stroma where there is low concentration of hydrogen ion, and this one are the two photosystem. These are the photosystem 2 and this one is the photosystem 1. So splitting of water molecule takes place in the photosystem 2. An oxygen molecule is generated along with hydrogen ions. And these move through cyclic phosphorylation to produce ATP from ADP. And these ATP are gone to the Calvin cycle for the production of sugar molecule. And if it goes into non-cyclic phosphorylation, then it passes through photosystem 1 through ferrodoxin through NADP plus reductase to form NADPH. So NADPH and ATP go to Calvin cycle where sugar molecule is synthesized. So the Calvin cycle uses ATP and NADPH to convert carbon dioxide to sugar. So the Calvin cycle, like citric acid cycle, regenerates its starting material after molecule enters and leave the cycle. The cycle builds sugar from smaller molecule by using ATP and reducing power of electrons carried by NADPH. तो यहाँ पे शुगर मॉलिक्यूल्स का बिल्डअप स्टार्ट होता है by using ATP and reducing power of electrons carried by NADPH. So carbon enters the cycle as CO2 and leaves as a sugar named glyceraldehyde 3-phosphate. For net synthesis of one molecule of glyceraldehyde 3-phosphate, the cycle must take place 3 times, 6 in 3 molecules of CO2. तो कैल्विन साइकिल के 3 फेजेस होते हैं। एक होता है कार्बन फिक्सेशन, जहाँ पे इट इज़ कैटलाइज्ड बाय रूबिस्को एंजाइम, देन देयर इज़ अ रिडक्शन, एंड देन देयर इज़ द रीजनरेशन ऑफ द CO2 एक्सेप्टेड बाय RUBP। तो 3 स्टेप्स होते हैं कैल्विन साइकिल में: कार्बन फिक्सेशन, रिडक्शन एंड रीजनरेशन ऑफ CO2 एक्सेप्टर। So over here CO2 enters in the presence of Rubisco, RUBP carboxylase. This one is the step 1 of carbon fixation, where 3-phosphoglycerate and 1,3-biphosphoglycerate is synthesized, which ultimately gets converted into 3-phosphoglycerate, and this is the second phase, which is the reduction phase, in which glyceraldehyde 3-phosphate is converted into is reduced, and this is the phase 3 where regeneration of CO2 acceptor RUBP takes place and ATP is synthesized. These are the 3 phases. This one is the phase 1 of carbon fixation. This one is the phase 2 of reduction. This one is the phase 3 of regeneration of the CO2 acceptor. और आउटपुट मॉलिक्यूल यहाँ पे ग्लिसरॉल्डिहाइड 3-फॉस्फेट, जो कि एक शुगर मॉलिक्यूल है, वो बन जाता है, दैट इज कन्वर्टेड टू ग्लूकोज एंड अदर ऑर्गेनिक कंपाउंड्स। एंड third phase is the regeneration of the CO2 acceptor, that is RUBP, ribulose biphosphate. So this is how CO2 is fixed into generating a sugar molecule as an output, and RUBP is regenerated again. So there are some alternative mechanisms of carbon fixation that have evolved in hot arid climates. So dehydration is a problem for plants, sometimes requiring trade-off with other metabolic processes, especially photosynthesis. On hot dry days, plant close stomata, which conserves water but also limits photosynthesis. The closing of stomata reduces access to CO2 and causes O2 to build up. These conditions favor a seemingly wasteful process called photorespiration. So photorespiration is an evolutionary relic in most plant C3. Initial fixation of CO2 takes place via ribosco, forming a 3-carbon compound. In photorespiration, ribosco adds oxygen instead of CO2. In the Calvin cycle, photorespiration consumes oxygen and organic fuel and releases CO2 without producing ATP or the sugar. So photorespiration may be an evolutionary relic because rubisco first evolved at a time when the atmosphere had far less oxygen and more CO2. So photorespiration limits damaging product of light reaction that build up in the absence of Calvin cycle. In many plants, photorespiration is a problem because on hot dry day it can drain as much as 50% of the carbon fixed by the Calvin cycle. Then there are C4 plants that minimize the cost of photorespiration by incorporating CO2 into 4 carbon compounds in the mesophyll cells, and this step requires the enzyme PEP carboxylase. So PEP carboxylase has higher affinity for CO2 than Rubisco does, so it can fix CO2 even when CO2 concentrations are low. These 4 carbon compounds are exported to bundle sheet cells where they release CO2 that is used in the Calvin cycle. तो पेप कार्बोक्सिलेज के पास हायर एफिनिटी होती है कार्बन डाइऑक्साइड की देन रूबिस्को does. It can fix CO2 even when CO2 concentrations are low. And these 4 carbon compounds are exported to bundle sheet cell where they release CO2 that is used in the Calvin cycle. So this is the C4 pathway. This is the C4 leaf anatomy. These are the mesophyll cells. And these are the bundle sheath cells. This is the vein, that is the vascular tissue. This is the C4 pathway where PEP carboxylase is converted into, alongside with PEP, CO2 PEP carboxylase converts into oxaloacetate and malate, which enters the bundle sheath cell from the mesophyll cell, and it is converted into pyruvate and CO2, and CO2 enters the Calvin cycle, which ultimately leads to the formation of sugar molecule, and it is distributed into the vascular tissue. So this is how C4 pathway takes place. This one is a C4 leaf anatomy once again. This one is a mesophyll cell. This one is the mesophyll cells. ये बंडल शीथ सेल्स हैं. और ये वेन्स हैं. तो C4 पाथवे में यहाँ पर पेप कार्बोक्सिलेज यूज होगा. जो कि कार्बन डाइऑक्साइड को यूज करेगा और कन्वर्ट करेगा इनटू ऑक्सैलोएसीटेट and into malate, which is converted into CO2 and pyruvate in the bundle sheath cells. CO2 enters the Calvin cycle, which is and gets converted into sugar, which is transported to vascular tissue. Then there are CAM plants, CM plants, that use cressulation acid metabolism to fix carbon. So CAM plants open their stomata at night, incorporating CO2 into organic acids. Stomata close during the day, and CO2 is released from organic acid and used in the Calvin cycle. This is the example of C4 plant, which is in sugarcane is an example of C4 plant, जहाँ पे mesophyll cells में CO2 यूज़ होगा और ऑर्गेनिक एसिड में कन्वर्ट होगा और अल्टीमेटली वो CO2 में कन्वर्ट होगा, जो कि बंडल शीथ सेल्स में एंटर करेगी और CO2 फाइनली कन्वर्ट होगी शुगर मॉलिक्यूल में. by use of Calvin cycle. In the case of CAM plants, it takes place during the night, where CO2 enters and it is converted into organic acid, which is ultimately converted to CO2 during the day, which is ultimately converted into sugar molecule by the help of Calvin cycle. And CO2 is incorporated into 4 carbon organic acids by undergoing carbon fixation, and organic acid release CO2 to the Calvin cycle. So what is the importance of photosynthesis? The energy that is entered the chloroplast as sunlight gets stored as a chemical energy in organic compounds. So sugar is made in the chloroplast. It supplies the chemical energy and carbon skeletons to synthesize the organic molecules of the cells. And the plants store excess sugar as starch in structures such as root, tuber, seed, fruits. In addition to food production, photosynthesis produces oxygen in our atmosphere. So this is an important process. तो थैंक यू गाइस. अगर आप लोग के कोई क्वेश्चंस हैं तो आप उन लोग उसे कमेंट बॉक्स में पोस्ट कर सकते हैं, एंड आई विल बी आंसरिंग योर डाउट्स एंड क्वेरीज. So all those doubts you are having, you can please post in the comment box and I will be able to answer them. And guys, please like, share and subscribe these videos, these YouTube lectures, and share with the people as much as possible. You can use my referral code HY10 to get 10% off on the next Unacademy subscription. तो अगर आप लोग के पास कुछ डाउट्स है तो उसको आप लोग पोस्ट करें कमेंट बॉक्स में. So I hope there are no doubts. You guys must have learned about chloroplast in this lecture and how photosynthesis takes place. Thank you guys